Hi, my name is Chloe,
And Logan.
and we're going to tell you about our research titled Ethogram Development for Trichoplusia. Introduction. Trichoplusia, or teeny, is a species of lepidopterans commonly known as cabbage loopers. Our work involves developing a controlled environment to study behavior that can help illustrate the impacts of environmental cues, and the neurological basis of these behaviors can be further investigated. Because of the complexity of behavior, the use of model organisms is essential in understanding biological phenomena, as they are easy to care for in a lab environment and have a short generation time. Teeny are easy to rear in a lab, and much is known about their life cycle, husbandry, and ability to survive in captivity, making them an excellent candidate as a model organism. Additionally, the study of teeny's simple neurological system can help us understand more complex neurological systems. This species exhibits 5 larval instars, with the 5th instar being the latest stage of development and the 1st instar being the earliest stage of development. Interestingly, teeny has no existing ethogram that we are aware of, and our goal is to create an ethogram that we can use in future research endeavors. An ethogram is a laundry list of behaviors and the descriptions associated with those behaviors.
Methods. The research population is reared using the same methods as the general population, with the only exception being staging every day. Staging involves monitoring and measuring the head capsule width of caterpillars to determine which instar they're in. Head capsule width is the best way to determine instars, as head capsule remain constant throughout each instar and only change when the individual molts and reaches its next instar. Once the individuals in the research population reach their day 1/5 instar phase, they're recorded in an arena, as seen in Figure 2, and their behavior is studied. Figure 1 shows a pictogram of a day 1/5 instar individual. All 4 edges of the arena contain a thin layer of Vaseline that is applied using a sterile cotton swab. Note the red arrow in Figure 2. Vaseline prevents individuals from crawling out of the arena. The process of ethogram development involves the study of 2 behavioral paradigms, each represented in Figure 3. Additionally, several variables will be tested in each paradigm, and these are also detailed in Figure 3, as well as how the setup inside the arena will occur. Each video is recorded for 30 minutes, and our current plan is to only record day 1/5 instars. Once the videos are recorded, they're coded by our team of researchers. Coding involves a group of people watching each video and noticing behaviors exhibited by individuals. Eventually, each of our paradigms will have an associated ethogram that will detail the behavior exhibited by individuals in that environment. We plan on determining the underlying mechanisms of behavior that can help inform our understanding of behavior.
Results. We have taken recordings for a single individual in an arena alone, without food, as well as 2 individuals in an arena together without food. We are at the beginning stages of our coding, but so far we have noticed that the major behaviors exhibited, this was determined based on the frequency of the behavior, are inchworm, searching, and wall engagement. These behaviors, along with other behaviors we have coded for, are described in Figure 4. Figures 5 through 7 illustrate the frequency of behavior for both single and double paradigms, with error bars that indicate the standard error of mean. There were 3 replicates of the standard error of mean calculation. The graphs show the frequency of the behavior on the y-axis, and the x-axis represents time intervals over 30 minutes. We binned our data into 5-minute intervals, and this is represented in our bar graphs. Figures 5, 6, and 7 differ on the behavior reflected, and graphs A indicate the behavior of single individuals, while graphs B indicate the frequency of behavior in videos with doubles. It's clear from our data that individuals in an arena alone exhibit greater frequency of behavior than individuals in an arena together. Figure 8 shows a table that we have set up to run an inter-rater reliability test, comparing the coding results of Rater 1 and Rater 2. We plan to run this test for each video coded for each paradigm. Figure 8 only illustrates an example of one of our videos for a single individual in an arena alone without food.
Future directions. Our future directions include continuing to collect videos and data for each paradigm to further develop our ethogram. We also plan to add food to the arena and note how behavior changes based on the addition of this new variable. Once our ethogram has been developed, our plan is to compare behaviors between different instars and possibly even compare behaviors between parasitized and non-parasitized teeny.
Thank you.